Well, hi everyone. I am really glad to be talking to you all. I'm a professor over at Stellenbosch University, just the Tygerberg campus, very close by. So I try to show up here as often as I can, at least every 2 weeks on Thursdays. So if you see me around and need to ask a question, I'm available. I've been really glad to be working with some of the data sets that you're producing here, and we're trying to improve the way we identify some peptides and proteins in plants, which has not classically been an area where we've seen an awful lot of proteomics, in large part because the genome projects for plants can be so much more difficult than they are for other species. But today, we're going to talk about algorithms. I realize that you may be coming to this lecture with a bunch of bench biology background and no computer science training. It may be that you were a mathematician in your high school years but haven't really touched it lately. I'm not going to try to hammer you with a whole bunch of equations, but try to make peptide identification algorithms just as clear as we can. So with that said, let's talk about peptide identification from tandem mass spectrometry. Okay, so we're going to start with a little bit of analytical chemistry. Analytical chemistry is not where I came from. I got my training in a biotechnology department at the University of Washington. But analytical chemistry is very important for us to understand because this is the branch of science that produces the spectra upon which most proteomics is based. So we'll be talking about the problems of collision-induced dissociation, CID. This is the technique most commonly used to go from a peptide to a tandem mass spectrum. We'll talk about that in some detail. And then from there, we'll talk about the structure of database search identifiers. These are the algorithms that we use to make sense of tandem mass spectra. And from there, we'll talk a little bit about how we do scoring, how we can figure out whether a peptide is truly represented by a tandem mass spectrum or not. All right, now this diagram is busy, but it's got an awful lot of pieces that we really need to understand to get the dominant paradigm, the standard technique by which we identify peptides in mind. So we start with a protein mixture. How complex is a complex protein mixture? Very open-ended question. Okay, so somebody might hand you a pull down. They might have done an immunoprecipitation, in which case they pulled down a protein by affinity, and they want to see what proteins does it associate with. Does it come down with other things? That's a relatively simple mixture for proteomics. They might have given you just a total cell lysate to work from. Total cell lysates are hideously complex. How many genes do we have in our cells? Anyone know? The number changes every year, it seems, but there's kind of a current number how about a ballpark figure? How many genes does a human being have? 3.2 billion? That's nucleotides. Our total genome spans a huge number of nucleotides, but the number of genes is much, much smaller than that.
50,000?
50,000. That's an overshoot. 25? 25 is a little higher than most people go these days, but it's quite close, actually. I think the number is somewhere between 20 and 22,000. So how many genes, by distinction, does an E. coli rely upon? Just over 4,000. A yeast has 6,000. So to be a human only requires about 3 times as much genetic material as it does for yeast, not counting all the regulatory stuff. Okay? So that's the size of a human proteome. Now, does that mean that all 20,000 of those genes are producing transcripts wildly in every cell? No. That's why proteomics gets to be so much more complicated than genomics, because proteomes are differential by cell type, and even by the condition of stimulation. So what's that? That's certainly the environment, yes. If you drink, we call that the orange juice problem. If somebody drank orange juice that morning, their proteome is different. It's okay. All right, so that's a complex sample. If you have 20,000 genes that are all producing multiple transcripts, you may have 70,000 different proteins that you have to consider as possibilities whenever you're looking at a protein sample. And if you're dealing with a cell lysate, you'll probably have at least 10,000 distinct proteins that you could plausibly identify from that sample. So that's a lot of complexity. So why would it be then that the very first step in this biochemical workbench method, we make the mixture more complex? If we started with 10,000 proteins, maybe 20,000, I mean, how many proteins we actually have, we don't really know because these things just keep going. We have those we can see and those below the limit of detection, basically. So the first thing we do then is to digest up all of those proteins into peptides. This involves denaturing them, it involves reducing them because those disulfide bonds that they have within their structures can really block identification. And we digest them using an enzyme called trypsin. Trypsin cuts where? Has anyone heard of trypsin? Good, okay, so trypsin cuts where? Arginine and lysine, okay, that's great. The two most basic amino acids. So that means that we're going to produce maybe 10 times as many peptides as we had proteins to start with. Then we're probably going to use some sort of fractionation method. One of the most common ways that you see out there is to separate that proteome on a 1D gel, and then those 1D gel slices each get handled as a different fraction from the same sample. You also see people using techniques like mud pit or basic reverse phase liquid chromatography or Hillick columns. There's any number of ways. But this fractionation method is one of the ways that we separate one sample into many that each represent subfractions of that proteome. Then we use liquid chromatography. Liquid chromatography uses very hydrophobic beads. So basically the peptides stick to them with different levels of affinity. Those that are most hydrophobic, those that are most greasy, basically stick really well, and those that are quite polar don't stick as well. So over the space of time, over the space of maybe an hour of a shallow gradient, as we gradually increase the hydrophobicity of solvent flowing through it, different peptides come off of that column and flow out into electrospray ionization. Electrospray ionization is one of the two technologies that led to a recent Nobel Prize in the field of proteomics, because electrospray ionization is a great way to produce ions from biological compounds, in this case peptides. So in effect what we're doing is sticking a cattle prod on the end of a water hose. We have the peptides coming off of the column, and we're charging them up to 3,000 volts, and suddenly we get this spray of ions appearing. So each of these peptides may take on 1, 2, or 3, or 4 protons, which makes them ionize. And then we have the mass spectrometry side. Now there are two things I really want you to get about the measurement of peptide ions in tandem mass spectrometry. And that's that we measure them twice. The first time, we're generally doing some sort of high-resolution mass spectrometry. In this case, I've drawn what's called an orbitrap. It's a very common kind of analyzer that we see used in proteomics. And an orbitrap gives us the ability to do a Fourier transform-based method for getting very, very accurate masses for the peptide ions in intact form. Before we've done anything to them, we're just looking at what ions are present at this particular moment in time. This is called a mass spectrum. So you end up with an M/Z axis and an intensity axis. And if you see a big spike pointing up at a particular M/Z, that tells you that there is an awful lot of some ion at this mass to charge value. Now I'm going to talk about mass and mass to charge both in this talk. And it's important to understand that the mass analyzer, the device that's actually doing all the detection that's doing all the manipulation in these experiments, is always manipulating and measuring the ions on the basis of mass to charge ratio. So the ions are taking on some number of protons. That number of protons tells us the charge that it takes on. And when you deal with something like an orbitrap mass analyzer, you've got resolution to spare, which means that even if these things are charged at +5 or +6, or for that matter +20, you can still tell the difference in mass to charge value between the different isotopes of the peptide. A single neutron is clear as night and day in a Fourier transform mass analyzer. Okay, so that's very important. So we have to begin with, and if you can tell the spacing of isotopes for an ion, you also know its charge. So having measured the mass to charge, you get the mass because you can observe the charge just by spacing of isotopes. So mass analyzers have come a very, very long way in the time I've been in this field, and it's astonishing to me how much better our measurements are today than they used to be. All right, so that's measurement number 1. This is just kind of a parts list. What's visible for what peptides are currently leaving the column that are visible now? That's a mass spectrum. And the instrument in data-dependent acquisition, one of the most commonly used technologies in the field, is to, the instrument prioritizes which of these ions will be isolated, bounced off of gas molecules to create fragments, and then measured in a tandem mass spectrum. So if it sees a whole bunch of major ions in this mass spectrum, it will probably add those to the list to be isolated, fragmented, and measured in tandem mass spectra. So this tandem mass spectrum, then, is different than the mass spectrum we collected over here. It's still measuring things in mass to charge and intensity values, but now it is reporting not what ions are leaving the column at this point in time, but rather what are the fragments produced when we explode one of those. Right? Okay, so the tandem mass spectra then are evidence associated with individual peptides, and the mass spectra are just kind of our guide to what peptide masses led to these tandem mass spectra. All right, are there any questions before we move ahead? Because this is all, this is the framework into which everything fits. Okay. So, there are several places in which we're disrupting the peptides here. So I wanted to make sure that we keep track of which things are on the bench, which things are happening in the mass spec, which things are happening in the column, and so on. So these are disruption steps. So we start with denaturation and reduction. We talk about proteins as having different levels of structure. Does anyone remember what those are? Right, okay, so primary, secondary, okay, right. So we start with the chain of amino acids itself, connected together by amide bonds, those are the primary structure. We've got secondary structure, which are localized structure like alpha helices and beta sheets that are formed by hydrogen bonds, blah, 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 blah. Okay, so we need to get rid of that structure, and we're going to do it through denaturation and reduction. Generally speaking, we just use urea. If you have a high enough concentration of urea, proteins start stretching out. And we use reduction by generally dithiothiazole, DTT, to just break apart those disulfide bonds. I think of those sort of like safety pins holding the structure in place. So you gotta snap those. All right, but that still leaves the backbone intact for these things. Enzymatic cleavage, on the other hand, is what digests the protein into shorter peptides. And because we're using trypsin, most typically, we will cut after arginine and lysine residues, and that hopefully is going to give us a bunch of peptides that are something like 6 to 30 amino acids in length. That would really be ideal. If it's much bigger than that, we can't identify it as successfully. And if it's smaller than that, it doesn't give us a whole lot of information. I mean, think about it. If you've got 3 amino acids, those could be associated with any number of proteins in the database. Okay, and then CID, this collision-induced dissociation, is the process of fragmenting a peptide ion through collision-induced dissociation. We're going to spend a couple minutes talking about that. Okay, so here's our cartoon down at the bottom. I've got my peptides floating around in space, and they're colliding with gas molecules. So we start by saying that mass analyzers work best when they're kept in a vacuum. So it might seem odd that we would pump gas into these mass analyzers. And one of the reasons that we do this, in quadrupole ion traps at least, is to give these peptide ions something to bounce off of, which makes them a little easier to trap to begin with, and enables us to add energy to them. So for this, I want you to think of walking down a busy sidewalk in Johannesburg at rush hour, right? It's a busy day, people are all in a great mass, there are people everywhere, and you might bump into people. But no one dies, generally speaking, from bumping into somebody on the sidewalk. What happens, though, if you try dashing down the sidewalk in a case like that? Suddenly these bumps are not so innocuous, and sooner or later you're going to break an arm or a leg. Peptide ions are the same. They're moving around in the trap in these nice lazy circles, and that's all just fine. But suddenly, when you punch them up with a powerful bunch of energy, you see them start oscillating a lot more quickly within the trap, and these collisions with gas molecules are happening at a much faster rate, and sooner or later, the temperature of these peptide ions rises. And some weird things start happening there. So let's talk about that. All right, at the left, we have our peptide ion in intact form. This is 4 amino acids, so is anyone a Star Trek fan here? No one's a Star Trek? We have a couple Star Trek fans. That's good, that's promising, okay. So, does anyone know the call sign of the USS Enterprise? Well, all right. It's the NCC 1701. All right? But NCC is your clue, because NCC tells you everything you need to know about the backbone of peptide ions. So you see at the left we've got an N, that's at the N terminus of the peptide, and it's called the N because of that amide over there. And then it's connected to an alpha carbon, and that's attached to a carbonyl. NCC, there it is. Right, so we've got NCC, NCC, NCC. It's like a waltz too. That's all right. So you see, I've got a little thing that looks like a little blur there on the second OH there, right? That is a proton. The proton has decided it's comfortable at this location. Now, normally speaking, if you've got a peptide sitting in nice lazy circles inside the mass spec, it's quite content to hold that proton on the lysine or the arginine, because those are very basic amino acids by comparison. However, I just said we were adding temperature to this ion. It's in a vacuum, so how do you have temperature in a vacuum? Well, you start sampling new conformations, and things that can move around, like protons, start sampling new locations. So in this case, this one has sat down on a carbonyl to form this OH. And now we see that the electrons on that very first carbonyl get a little excited. They say to themselves, my goodness, you see the proton that's sitting on that carbonyl is kind of scooching the electrons up towards the O, because now it's a little more positive than it might be, which means that the carbon of that carbonyl has become a little bit, it's feeling a little needy, frankly. It wants some electrons around it. So the electrons on that O of the first carbonyl start attacking in that direction, and suddenly you get a cyclization like this. And the cyclization results in a breakage of this 4 amino acid peptide into a pair of 2 amino acid fragments. And because this is a break at a peptide bond, we call the one on the left a B ion and the one on the right a Y ion. So this is the common nomenclature that we use for it. If it broke elsewhere, we would say it creates an A and an X, or a C and a Z ion. But when it breaks at a peptide bond, we call it a B and a Y ion. Okay, so this is what has happened. The proton has moved to a place that causes a shift in the electronics of the peptide. We get this attack across it, and we get a cyclic B ion and a Y ion that's just kind of like a smaller peptide in form. Okay, and that is what produces tandem mass spectra, because any given peptide will have lots of peptide ions at which lots of peptide bonds at which it might break. And if you have 10,000 copies of this ion isolated in the trap, all undergoing CIE at the same time, some will break between the second and the third amino acid, some will break between the third and the fourth, or the fourth and the fifth. They all break in different places. So in this case, we have 10 amino acids in this peptide. And I've shown a break after which amino acid? The number 4, okay. So we call the ion on the left the B4 ion, because it's an ion of B type, it includes the N terminus, and it has 4 amino acids in it. And then we have this other one, and I already said it was called a Y, because it's the C terminus. But it has how many amino acids? Okay, so that's a Y6 ion. All right, now I could make the joke that because this is the B4 ion, this is the after ion, but you wouldn't laugh. All right, so here we go. Now we have measurements of the fragment ions. These are each the M over Z values. This is an actual tandem mass spectrum collected for this peptide. And we see that we have this ion at 572 that represents GTIGPK, and we have 415.2 that represents the first 4 amino acids. So as you go from the N terminus over to the C terminus, those B ions keep getting bigger and bigger, which means that they appear in this order, low to high, on the tandem mass spectrum. But as you go from the N terminus to the C terminus, the Y ions keep getting smaller, which means that the most N-terminal Y ion is over here, and the least is over there. All right, so we have two series of ions that tell us the sequence, but they're reversed with respect to each other, like that, and overlaid on each other. If you look at this spectrum, would you feel comfortable inferring the sequence just by looking at the spectrum? I've done this a few times, and it's really painful and awful, and I'm not very good at it. I need software to help me do this, and that's why we have database search algorithms. Okay, so on the bench, what you've done is to start with proteins, digest them to peptides, and then produce tandem mass spectra that correspond to those peptides. informatically, we now need to reverse all of those steps. Starting from the tandem mass spectra, we need to put together our best guesses of what each spectrum represents in terms of a peptide sequence, figure out which of those identifications, those putative identifications, we can actually trust, and then we need to infer a set of proteins that explain the peptides we've seen. Now this lecture focuses strictly on this problem. How do we get our best guess of what each spectrum represents? The other things I can talk about as well, I'm happy to, but we just have a relatively short period to talk about this. So we're going to focus on the problem of looking up the answer in the back of the book, doing a database search to identify tandem mass spectra. Any questions so far? Okay. We have another figure that's really important to understand for getting the structure. This is probably the most important slide, the most focal slide of the entire talk. This slide explains the steps that we undertake to transform a spectrum into a peptide spectrum match, a PSM. A PSM represents one peptide associated with one tandem mass spectrum and an alignment of one on the other. So we're going to start with a search space. Search space is a really big concept that shows up again and again in the field of bioinformatics. It means that we need to tell the software what's permissible, what rules are we using to describe what proteins we could possibly identify here. And a FASTA protein sequence database is our source of all the proteins that will be considered. If you are searching a human sample, you are not going to give it Saccharomyces cerevisiae as a search space, right? If it's the database, and this is a very important rule, database search tools, database search identifiers in proteomics, will only recognize those proteins that you supply as possible answers. So we have to have a sequence database, or this is totally off the board. This is just not going to work. If you don't have a list of the proteins that are present in that species, you're doomed from the get-go. Okay, so we have our set of protein sequences that are provided. Do anyone know how many sequences how many different species have been subjected to sequencing at this point? It's a ludicrous number. When I was in grad school, we had just finished the yeast genome, and we were really proud of ourselves. But these days, more than 1,000 species have been identified. The other day, we were working with hyena data, and we were astonished to discover that hyena hadn't been sequenced. But I tell you, most everything out there has. Your cat, your dog, they've all been sequenced. So that's great. So okay, we start with these protein sequences. Now the software has to be told what kind of enzyme did you use to cut up your proteins. We use trypsin, so the software has to emulate the behavior of trypsin to guess which peptides would have been produced had you digested up these protein sequences. So in silico, it's doing the same thing that you did on the bench. Okay, so we now have these peptide sequences. Next, you need to tell the software which modifications, which post-translational modifications are possible within this data set. Now some of them we always look for. So the first of these would be methionine oxidation. Methionine normally has a mass of 131, but we allow it to be oxidized simply because your samples are exposed to air, and methionine sometimes gets a little grabby with the oxygen and gains a mass of 16. So 131 plus 16 is 147. All right. So that means every time that there's a methionine in the peptide, we have to kind of take a fork in the road. We have to consider that it might be unmodified or it might be modified. We have to consider both possibilities. So that is a PTM expansion. I'm saying that just relates to these peptides getting decorated. And then we have kind of the critical innovation that made database search work, which is the notion that we don't have to compare every spectrum to every peptide. That would be kind of ludicrous. Remember that we've measured these peptides twice. We measured their intact masses through mass spectrometry. So we only have to compare those sequences to a spectrum that match it in intact mass to a high degree of accuracy. All right, so those peptides for a particular spectrum that are close to it in intact mass are called candidate peptides. And for each of those, we're going to predict what spectrum it would produce if it were to shatter, and then the set of ions that we've predicted to appear in a tandem mass spectrum for this peptide may be compared to those that we truly observed in the tandem mass spectrum through a match scoring algorithm. Okay, so does that make sense to everybody? Very good, okay. Now, I would point out that even with an exquisitely accurate mass analyzer, we're still going to have multiple possibilities that get compared to every spectrum. If you're dealing with something like a quadrupole ion trap and a large proteome like the human proteome, you're quite likely to have tens of thousands of potential peptide sequences that could explain a given spectrum, which means that we're going to have a very good method for deciding which ones are the best matches and which ones are the poorest matches for a spectrum. We'll talk about how this works, actually about each of these steps as we move ahead. All right, so we start with this trypsin digestion function. It might seem like a very obvious thing to discern. If you've got a protein sequence, you just lop it every time you see a K or an R, and that's the set of peptides that could result from it. But in fact, there are some subtleties to this that make this a bit messier. So at the top, I've drawn in a protein, and I've just put in a gap everywhere that there's an arginine or a lysine. So we start with the fully tryptic peptides. These are the ones that have a lysine or arginine before you get to that sequence, so in order to produce the N-terminus of this peptide, and they end at a lysine or arginine. So we've chopped after that. So fully tryptic peptides are the ones that we get if we just allow every lysine and arginine to be cut. However, sometimes when we have a whole lot of a protein in a sample, for example, we will see a peptide that starts at a trypsin cut site but ends somewhere you don't expect, just at some amino acid residue, other than lysine or arginine. You wouldn't think that would happen, but in fact, many of our samples contain native proteases. If you're dealing with a whole cell lysate, some of these whole cell lysates have proteases of their own, other than trypsin, and some of them just keep going on and chopping things. So some of them end up producing semi-tryptic peptides. You may also have cases where a lysine or arginine was present in the sequence, but it didn't get cut by trypsin. So you have to take all of these into account when you're deciding what search space you will look for. Some search engines will default to finding only fully tryptic peptides and can miss that there's a broader set of peptides in there for you to identify. Okay, and certainly there are some cases where you have a protein that's got a long stretch of sequence with no arginines and no lysines. I think about this as like a transmembrane domain. If you've got a protein that extends through the membrane, is it going to have a whole bunch of lysines and arginines in a region that stretches through the membrane? No, because that's a really hydrophobic environment, and lysines and arginines are kind of polar in nature. So certainly some proteins have long stretches of sequence where there are no cuts made. Okay, now let us talk about this problem of post-translational modifications. The original sequest algorithm that we use for a lot of identification was published in 1994. But the original sequest algorithm could not identify peptides that were modified. In this case, we have another paper right on the heels of that in 1995 that showed how to modify the software to search for modifications as well. So here we have a peptide from alpha casein, digestek. When we look for phosphorylations, we typically look at 3 amino acids. Does everyone know what they are? Okay, it's serine, threonine, and tyrosine, those 3. All right, now serine and threonine are very common amino acids. Tyrosine is less so. And of course, there are no tyrosines in this sequence. Tyrosine is Y. Y is the other one. So every time we encounter a modifiable residue in this sequence, we have a fork in the road. So D, I, G, those are always going to be unmodified because this search space does not define any PTMs that take place on D, I, or G. But when we encounter S, we suddenly have a fork in the road. We have to consider all possibilities where the S is modified and all possibilities where the S is not modified. Further, once we get 2 letters down to ES, we have another fork in the road. Right after that S, we have another fork in the road, a T. And so you have to consider the cases where you went left, 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 or left, left, right, or left, right, left. It's a lot of possibilities. In this case, we have 3 modifiable residues, each of them having two ways that we could possibly go from it. So we say there are 2 to the 3rd possible paths through this peptide, which is 8. All right, so we have 1 possibility that's not modified at all, 1 possibility that's modified everywhere, 3 that are modified in 1 place, and 3 that are modified in 2 places. So this is why when we search for post-translational modifications, our search space expands exponentially, 2 to the 3rd, remember? So the more dense the modification sites are within a peptide, the higher that exponent, and the more possibilities you have to consider in mass for an individual amino acid, the higher the base of it is. So this is why post-translational modification searching can be very expensive. You don't want to use database search to look for acetylation and oxidation and phosphorylation and ubiquitination and so on, because your search will just never finish. It will also cut very badly into the sensitivity. So this is a problematic area for us. Okay, now the peptide mass filter refers to the accuracy with which you know the mass of each peptide. So what kind of mass analyzer do you use here? Kind of a long plastic box over there. I've seen it. Have y'all seen it? Big long plastic box. All right.
Have you not seen it?
What's that?
No, have you not seen it?
You haven't seen it? Well, you guys need to go on a tour then. You have seen it, okay. This is called a TOF mass analyzer. And TOF stands for time of flight. Okay? So your time of flight mass analyzer basically gives the ions a big kick at one end of the flight tube, and it times when ions arrive at the other end of that flight tube. And based on that flight time, it discerns what the mass to charge value is. So a time of flight mass analyzer sits somewhere in the middle of the mass analyzers we have available to us. Kind of the venerable and still widely used mass analyzer, the quadrupole ion trap, has what we call unit resolution. We can tell within a mass unit how heavy something is as it moves around in a quadrupole ion trap. In a time of flight mass analyzer, we're sitting somewhere in the middle. A really, really good time of flight can give you 10 ppm mass accuracy, meaning that you know the mass of an ion is within 10 parts per million. If you think about that, if you divide something that has a mass of about 1,000 by 1,000,000 and multiply that by 10, that's how accurately you know it. That's astonishing. Generally speaking, when I'm searching time of flight data, I usually assume a wider mass accuracy than that and allow for up to 100 ppm mass error. If I'm measuring something from one of these Fourier transform instruments that I mentioned, I usually assume that it has, say, a 10 or 20 ppm mass accuracy. Okay, so when we have a mass measurement, we see something that looks like this. We have an observed mass, but we know it's probably not exactly right, not to 0.00001, right? So we need to allow for some amount of error, and the wider we set that error, the more peptides we'll have to consider as possibilities for explaining the spectrum. So we want to have that mass tolerance set as narrowly as we can, but what happens if we set it too narrowly? What if our measurements are accurate to within 50 ppm, and we specify that it has to be accurate within 10 ppm? The problem here is that we're going to be considering peptide ions, we're going to be considering an incomplete set of peptide ions that might explain a given tandem mass spectrum. We'll still identify those that have extremely good mass accuracy, just by random chance. But we're going to lose a lot of sensitivity because we'll be excluding from consideration some peptide sequences that are close but not close enough to be within the mass tolerance. A lot of software also will have a feature for isotope correction. This happens when the instrument may make a mistake. It may see a set of ions like this in the mass spectrum and mistakenly select this as the monoisotope. So the instrument is reporting the wrong isotope's mass in association with this tandem mass spectrum. For something like that, we need to use isotope correction within the software. Okay, so that's a little discussion of the peptide mass filter, and now we get to spectral modeling. How do we predict what spectrum should occur for a given peptide sequence? When we have a +1 or a +2 peptide, our answers are pretty straightforward. They fall in this top category. We have, say, 2 peptide ions that are sitting on this peptide. This peptide has now broken. So there are really several possibilities. We could have both protons going with the N-terminus, so this goes away as an uncharged neutral. We could have the charge separating so that both carry off 1 proton, or we could have the case where just the C-terminus walks off with both protons. So, because of charge repulsion in a +2 peptide, generally speaking, both protons, the protons part ways, and each goes off with one of the fragment ions. So you end up with this top scenario. That's how software models that these +2 peptides will break. When you have a +3, on the other hand, things are a little weird. Now you have 3 protons going this. It's a little. If you have 2 housemates and they're parting up so that they're moving to individual places, more or less everyone can figure out whose was that blender, right? Once you've got 3 people involved, things get a lot more confusing. So here you've got a peptide that's breaking, you've got 3 protons sitting on it, and 2 are going to go with one side and 1 is going to go with the other. But you could honestly have a case where all 3 protons go with one of the departing fragments. Generally speaking, the software that we use today figures that every peptide break in a +3 is going to have 4 ions resulting from it. A B that's singly charged, a B that's doubly charged, a Y that's singly charged, and a Y that's doubly charged. So twice as many fragments get put into the spectra for +3 peptides as do for +2 peptides. And generally speaking, when we deal with an instrument like a TOF, we're dealing with MALDI rather than electrospray. So we're dealing with a whole bunch of singly charged peptides. For them, we can model that the proton goes this way, and the proton may go this way, and we just model each of those events. Okay, on we go. Oops. Now we get to the more complex part of database search algorithms, and this, frankly, is where all of the research, essentially, in database search goes. When people are publishing new database search engines, it's very rare that they're considering parts of the algorithm outside how we determine whether this set of fragments is a match or not for this spectrum. So, one of the first algorithms out there was published back in 2004. Now, 2004 is some time ago. That's 12 years. So, do you remember what kind of computers were available in 1994, not 2004. Sorry. 1994. Everyone has very clear and distinct memories of 1994?
Average about 2 years old. So.
About 2 years old? Did your families have computers in 1994? Ah, all right. See, I got my first computer in 1981. So 1981 was a long, long time back. All right, that's fine. So in 1994, I was in college, and I had just bought, I think, a Pentium computer. The Pentiums had just come out. And by the time I joined the lab in 1996, they were using a Pentium 2. Pentium Pro class computers, right? Yeah, Pentium 4 was a considerably faster machine. All right, so computers were not that fast at the time. And so we had to wait a long time. When we did an experiment on an LCQ ion trap back then, this is one of the first ion traps that was commercialized, we would get perhaps 2,000 spectra produced by a 1-hour experiment. What a wealth of information, 2,000 spectra. So these 2,000 spectra would be subjected to database search. And luckily, we were just dealing with yeast, with about 6,200 proteins, I think. So not so bad. In that case, we needed the ability to very efficiently separate good matches from bad for each spectrum. And the technique that we used was something based on the Fourier transform called cross-correlation. I'm not going to explain fast Fourier transforms at the moment. They're mostly just a mathematical shortcut in this case. But I want to point out that I've given you 2 different citations for this cross-correlation scoring. One is from 2008 and one is from 1994. The original algorithm published in 1994 had a relatively slow a relatively slow implementation that required that we do Fourier transform on a whole lot of things. The same programmer came back to that code in 2008 and realized that he had left some inefficiency in the code. And by changing that one line of code, the software got 10 times faster. So we had 14 years where we were wasting CPU cycles for the same answer that we could have gotten much more quickly. It's very vexing. Stuff like this happens all the time. It's fun. So we have a lot of steps to follow through with in doing a cross-correlation. We start by normalizing the observed spectrum. We chop it up into 10 zones, and within each of those regions we say the tallest ion in that zone now has a height of 1, and everything else is scaled against that. It's one way to make the spectrum look a little bit more like the models we create for them. So next we need to generate a model spectrum for what the peptide should look like. This is pretty easy because we know where all the peptide bonds are. We know how much mass is to the left and to the right of each. And so we can calculate what the mass of the fragment ion should be. We also know what charges it should appear in, so we can model multiple charges, for example, in the case of +3 peptides. Then we can, within the Fourier transform space, essentially rub one spectrum past the other. You have the one you observed, and you have the one you've calculated, and you can rub them past each other. Now, at what we call tau 0, we have an ion at 500 in our observed spectrum be the ion at 500 in the predicted spectrum. That's no offset between the two. But as we increase those tau values, we see that they step apart from each other in the m over z space. And for each of those positions, we can compute a score. The score is going to be the product of intensities at aligned peaks. So what we see then is that at tau 0, we have a really tall spike that reflects a good alignment between the observed and predicted spectrum. And then as we get to higher tau values, we still get positive scores. There's still some random matching between the observed and predicted spectrum. And so those are computed into the correlation values there. So the cross-correlation score tells us how much better our spike was at tau 0 versus the tau values that we get from all this random matching. So this method is not really something that has an easy interpretation. You know, it's not a p-value that pops out of this. It's a score that says how much better is the alignment at tau 0 than at small values. And that is a little hard to interpret. But we have found that over the long haul, this scoring method has been really, really robust, especially for noisy spectra. So this is a method that we pulled out of mechanical engineering, and it has remained in use to today. However, there are many, 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 many search algorithms that have been published. I think at last count we were over 30 different search algorithms. And as I said, the way that most of them differ from each other is in how they do this final scoring. So I wanted to bring you this example as kind of a counter to the cross-correlation based method. Now for this, I really prefer having my big jar of marbles. I don't have it because it's sitting on my desk over Stellenbosch, but it's okay. I have a big jar of marbles. This jar of marbles represents the spectrum. Some of the marbles are white. They reflect places in the spectrum where there could have been a peak, but there wasn't a peak. And some of the marbles in the jar are black, a smaller number. And these black marbles represent places where we went looking for a peak, where there is a peak present in the spectrum. So I want you to think about this jar of marbles. Now I'm going to reach down into it. Now I've pulled out a subset of those marbles. Most of the marbles are still down in the jar. But the ones in my hands represent the peaks that were located at the position, the marbles that correspond to the positions I was looking in the spectrum for a matching peak. So I'm going to ignore for the moment all of the peaks and non-peaks in the spectrum for places that don't correspond to this peptide sequence. But I know that these locations, here, and here, maybe 30 locations, are places where there should be a peak, or would likely have a peak, if it represented this peptide sequence. So I've got those in my hands now. All right, so let us imagine that I scooped 20 marbles out of the jar. There were 1,000 marbles in the jar to begin with. 100 of them were black and 900 of them were white, which is to say that the original spectrum had 100 locations that did have a peak and 900 locations that didn't. Now in my hand, I scooped out marbles. 15 of them were black and 5 of them were white. In this case, I want to be able to say, how likely is it that by random chance alone, I would have scooped up this many black marbles? And the hypergeometric distribution gives us that answer. So this is a way to compute a statistic, a probability in this case, a direct probability, that this particular event would have happened by random chance alone. So this is what the math looks like. It's a lot of combinations and so on. And out pops the number 3.63 times 10 to the minus 12th. Not very likely to have occurred by random chance. But I want you to keep in mind that I'm going to be computing a score like that for every peptide compared to this spectrum. So just by random chance, if I compare 10,000 different sequences to this spectrum, I'm going to have an awful lot of scores. The chance that the best one has a score like this by random chance is somewhat better because I've done so many comparisons. All right, this doesn't look anything like cross-correlation, but both of them are equally useful in examining a spectrum and asking whether this peptide is a good match to it or not. All right, what happens typically in a database search? First off, we generally only care about the best scoring match. We may have 10,000 sequences we've compared to the spectrum, but generally speaking, we only ask ourselves, is the best match to this spectrum one that we trust or not? So that's quite a simplification. There are some cases where we see that we can look at the second best or the third best match, but generally speaking, we only care about the best ranked for each spectrum. Fewer than half of the spectra we collect from an experiment are likely to be identified. This is a hard fact for a lot of people in our field. This can happen for a lot of reasons. If the spectra represent a protein that is not in the database, there is no possibility that you will identify it correctly. If a spectrum represents a modification that you didn't say was possible, you have no possibility of identifying it. If your mass accuracy for the precursor was off, if this peptide just happened to have a really wild mass measurement, you're not really going to be able to identify it. So there are lots of things that can lead to this. Generally speaking, most mass analyzers that we use try to just maximize the number of spectra that we can accumulate for a particular sample. And that means they're digging down really far into the noise. Occasionally, there just aren't enough copies of a peptide to fragment well, informatively, to allow a confident identification. So this is one of the reasons, several of the reasons really, why we identify fewer than half of the tandem mass spectra that we generate in a shotgun experiment. Okay. If your MS scans come from a quadrupole ion trap, you probably won't be able to discern what its charge is. And this may mean that you end up trying to identify the same tandem mass spectrum once as a +2 and once as a +3. So in some cases, the identifications, you have more identifications than you have spectra to sort through. And of course, as I mentioned at the start of the talk, just because we have gone through this step, we haven't really identified proteins. We must go through PSM filtering, we must filter the peptide spectrum matches that we generate, and we need to infer proteins that explain the peptides that we've seen. So we'll talk about those steps in the hands-on tutorial. All right. So scoring differs quite a lot among different database search engines. But generally speaking, in every case, we are going to extrapolate a set of peptides from the list of possible proteins. We are going to predict what spectra they would produce if they fragmented, and then we compare those models of what the spectra should look like to the observed tandem mass spectra. Existing scorers basically tell you much more about the people who wrote them than they do about the best way to do this. There are lots of ways to say which spectrum is the best, which peptide is the best explanation for a spectrum. And we borrowed from people from engineering or mathematics or statistics. Our field has absorbed any number of people from physics, for that matter, who learned how to deal with mathematics quite well. So database search is ubiquitous for tandem mass spectrometry. In 9 out of 10 papers that you read in our field, you're going to see the use of a database search algorithm, whether it's Mascot or XTandem or SpectrumMill or any number. There's, as I said, more than 30 of these have been published to date. So this understanding how they work is very fundamental to working in our field.